Hey guys, welcome to part 2 of our episode on the electron transport chain and on mitochondrial disease. We've left off from part 1 where we last talked about uncoupling. If you haven't watched part 1, watch it because we're going to continue from there and it might not make sense. Okay, so, finishing on coupling, I want to start focusing more on the mitochondria and focusing on the disease part of the episode. So, if you're unfamiliar with the structure, I just ask questions to AI and AI tells us the answers and we take them with a pinch of salt. I hope you enjoy the episode. So, Grog, tell us about mitochondrial DNA. Let's start a bit with its structure so we understand what might be going wrong afterwards.
Mitochondrial DNA is a small, circular, double-stranded molecule in the mitochondrial matrix, about 16,569 base pairs long, encoding 37 genes, including 13 for electron transport chain proteins, 22 tRNAs, and 2 rRNAs. Unlike nuclear DNA, it lacks introns, is densely packed, and inherits modernly, as sperm mitochondria are typically degraded during fertilization. Its location in the matrix exposes it to reactive oxygen species from the electron transport chain, increasing mutation risk, which can lead to mitochondrial diseases.
So it's a circular double stranded molecule, and we've got 13 respiratory chain proteins. And what other functions does it have?
Mitochondrial DNA, mtDNA, encodes 13 proteins crucial for the electron transport chain complexes 1, 3, 5, and ATP synthase, plus 22 transfer RNAs, tRNAs, and 2 rRNAs that support mitochondrial protein synthesis. These tRNAs and rRNAs enable mitochondria to translate their own proteins, independent of nuclear DNA, to maintain energy production. Mutations in these genes can disrupt ATP synthesis or protein assembly, leading to mitochondrial diseases.
Okay, and let's talk a bit about maternal inheritance. So the embryo essentially gets all of its mitochondria from the egg, and I wonder how this is possible.
Mitochondrial DNA, mtDNA, is modernly inherited because the egg provides nearly all mitochondria to the embryo, while sperm mitochondria, which are few, are usually degraded after fertilization by a process called ubiquitin-mediated destruction. This ensures the embryo's mitochondria come from the mother, carrying her mtDNA, which is why mitochondrial diseases often trace through the Martol line. The egg's large mitochondrial population, around 100,000, compared to the sperm's few hundred, also overwhelms any paternal contribution.
So there is. So what impact does this have on humanity? The fact that we basically we only have um, we only have um, mother inheritance.
Maternal inheritance of mitochondrial DNA, mtDNA, means all humans inherit their mitochondria solely from their mother, shaping how mitochondrial diseases, caused by mtDNA mutations, are passed down exclusively through the maternal line. This impacts humanity by limiting genetic diversity in mtDNA, increasing the risk of inherited mitochondrial disorders like Leber's hereditary optic neuropathy in families with mutations. It also allows tracking of mitochondrial lineages and population genetics, but restricts paternal mitochondrial contributions.
And why did nature or evolution or whatever cause choose this?
Maternal inheritance of mitochondrial DNA, mtDNA, likely evolved because eggs contribute far more mitochondria, about 100,000, than sperm, a few hundred, making maternal dominance efficient and degrading sperm mitochondria prevents mixing that could disrupt mitochondrial function. This setup avoids potential conflicts between paternal and maternal mtDNA, ensuring stable energy production critical for early embryonic development.
And those mitochondria are in the tail, so they don't get absorbed basically on fertilization.
Exactly. Sperm mitochondria are mostly in the midpiece and tail, used for motility, and are typically degraded after fertilization by ubiquitin-mediated processes in the egg. This ensures only maternal mitochondria from the egg's cytoplasm are passed to the embryo, maintaining maternal inheritance of mtDNA. It's a neat evolutionary trick to avoid mitochondrial DNA conflicts and keep energy production consistent. Want to dive into how this ties to mitochondrial diseases?
I want to ask you, why does oxidative phosphorylation decline with efficiency decline with age?
Oxidative phosphorylation efficiency declines with age because mitochondrial DNA accumulates mutations from reactive oxygen species produced during electron transport, damaging ETC proteins and reducing ATP output. Aging also impairs mitochondrial biogenesis and dynamics, like fusion and fission, leading to fewer, less functional mitochondria. Additionally, declining antioxidant defenses allow more oxidative stress, further harming ETC complexes and membranes, especially in energy-demanding tissues.
Okay. And so there's a lot of mutations that accumulate too by reactive oxygen species.
Yeah, reactive oxygen species, ROS, produced during the electron transport chain, cause cumulative mitochondrial DNA, mtDNA mutations over time, especially in the matrix where mtDNA lacks robust repair mechanisms. These mutations disrupt ETC proteins and tRNAs, reducing oxidative phosphorylation efficiency and ATP production. This oxidative damage is a key factor in aging and mitochondrial diseases in high-energy tissues like the brain and muscles. Want to focus on specific mutations or their effects?
Tell me about defects in oxidative phosphorylation then. And why is oxidative phosphorylation? I don't understand what it is still, to be honest. So explain to me what it is, how it's linked to the conversation.
Oxidative phosphorylation is the process in mitochondria where the electron transport chain, ETC, uses electron flow from NADH and FADH2 to pump protons, creating a gradient that powers ATP synthase to make ATP from ADP and phosphate. Defects in oxidative phosphorylation, often from mtDNA mutations or damaged ETC complexes, reduce ATP production, leading to energy shortages in tissues like muscles and brain, causing mitochondrial diseases. These defects tie to our talk on mtDNA and aging, as mutations from reactive oxygen species disrupt the ETC, lowering the efficiency of this ATP generating process. Want more on specific disease symptoms?
I don't get it.
Okay, no worries. Oxidative phosphorylation is how mitochondria use oxygen and nutrients like NADH to create energy, ATP, by passing electrons through protein complexes in the electron transport chain, which pumps protons to drive
What's the difference between oxidative phosphorylation and the electron transport chain?
The electron transport chain, ETC, is a series of protein complexes in the inner mitochondrial membrane that transfers electrons from NADH and FADH2 to oxygen, pumping protons to create a gradient. Oxidative phosphorylation is the broader process that includes the ETC and ATP synthase, which uses the proton gradient to produce ATP from ADP and phosphate. Essentially, the ETC is the engine driving the proton gradient, while oxidative phosphorylation is the whole ATP making machine, including both the ETC and ATP synthase.
Okay. Let's talk a bit about different reactive oxygen species such as superoxide anion, hydroxyl radical.
Reactive oxygen species, ROS, like superoxide anion and hydroxyl radical, are highly reactive molecules formed as byproducts during the electron transport chain when electrons leak and react with oxygen. Superoxide anion, O2.9, forms at complexes 1 and 3, damaging mitochondrial DNA and proteins, while hydroxyl radicals, all OH, generated from superoxide via reactions like the Fenton reaction, are even more destructive, attacking lipids and enzymes. These ROS contribute to mitochondrial dysfunction and diseases by causing oxidative stress, especially when antioxidant defenses weaken. Want to dive into how these affect specific mitochondrial disorders?
Explain to me how the respiratory chain is a major producer of reactive species.
The electron transport chain, ETC, in the mitochondrial inner membrane produces reactive oxygen species, ROS, like superoxide anion, when electrons leak from complexes 1 and 3 and react with oxygen in the matrix or intermembrane space. This happens because the ETC handles high-energy electrons from NADH and FADH2, and small amounts escape the chain, especially under stress or damage, forming ROS. These ROS, including hydroxyl radicals formed from superoxide, damage mitochondrial DNA, proteins, and lipids, driving oxidative stress and mitochondrial dysfunction.
Okay, and how do these reactive oxygen species emerge, though?
Reactive oxygen species, ROS, emerge in the electron transport chain when electrons, normally passed from NADH or FADH2 through complexes 1 to 4, leak out, mainly at complexes 1 and 3, and react with oxygen in the mitochondrial matrix or intermembrane space, forming superoxide anion, O2.9. Superoxide can then convert to other ROS, like hydrogen peroxide or hydroxyl radicals, through chemical reactions like the Fenton reaction, especially when antioxidant defenses are low. These leaks happen more under stress, high metabolic demand, or mitochondrial damage, amplifying oxidative stress. Want to dig into how cells counteract this?
Sure. I also want to understand mitochondrial diseases now. So, start linking everything together. So, we have defects in mitochondrial enzymes and systems, in oxidative, and then we have, yeah. Tell me more.
Mitochondrial diseases result from defects in mitochondrial DNA or nuclear DNA, disrupting oxidative phosphorylation by impairing electron transport chain, ETC, complexes, or enzymes like ATP synthase, leading to reduced ATP production. Reactive oxygen species, ROS, generated during ETC electron leaks at complexes 1 and 3, cause mtDNA mutations, worsening enzyme dysfunction and energy deficits in high-energy tissues like the brain, heart, and muscles. These defects manifest as symptoms like muscle weakness, neurological issues, or heart problems, seen in disorders like MELLIS or Lee syndrome, with maternal mtDNA inheritance often driving their transmission. Want to focus on a specific disease or symptom?
I want to ask you about heterogeneity of mitochondrial diseases. So, we have homoplasmy and heteroplasmy. What does that mean? And how does it relate to genetic bottleneck and threshold effect?
Homoplasmy means all mitochondrial DNA, mtDNA, copies in a cell have the same sequence, either all normal or all mutated, while heteroplasmy means a mix of normal and mutated mtDNA, with disease severity depending on the mutant proportion. The genetic bottleneck during oogenesis reduces the number of mtDNA copies passed to the egg, amplifying heteroplasmy variability in offspring, as only a subset of maternal mtDNA is transmitted. The threshold effect means symptoms appear only when the proportion of mutated mtDNA exceeds a critical level, varying by tissue energy demands, explaining the diverse severity and symptoms in mitochondrial diseases, like MELIS. Want to dive deeper into specific diseases or these genetic mechanisms?
So I got genetic bottleneck, but why does genetic bottleneck occur and what's the threshold effect? Also, I didn't understand the word heterogeneity and homoplasmy.
The genetic bottleneck occurs during oogenesis when only a small number of maternal mitochondrial DNA, mtDNA copies are passed to the egg, randomly amplifying or reducing the proportion of mutated mtDNA in offspring, leading to variable disease expression. Heterogeneration in mitochondrial diseases refers to their diverse symptoms and severity due to varying levels of mutated mtDNA, while homoplasmy means all mtDNA copies are identical, all normal, or all mutated. The threshold effect is when symptoms of mitochondrial diseases like muscle weakness or seizures appear only if the proportion of mutated mtDNA in a tissue exceeds a critical level, typically 60-80%, depending on the tissue's energy needs. Want more on specific diseases or these terms?
Um, well, so in the threshold effect, we have normal mitochondria, we have dysfunctional mitochondria, and these are distributed unequally. They do not necessarily reflect the ratio. So maybe we just by accident get all the bad ones basically, and then this will be amplified.
Yeah, exactly. In the threshold effect, cells have a mix of normal and dysfunctional mitochondria due to heteroplasmy. An unequal distribution during cell division can randomly concentrate bad ones, amplifying disease severity if mutated mtDNA exceeds a critical level, 60-80%. The genetic bottleneck during oogenesis randomly selects a subset of maternal mtDNA, which may include mostly mutated copies, worsening the effect in offspring. This uneven distribution, not tied to age, explains why mitochondrial disease symptoms vary widely across tissues or generations. Want to dive into a specific example or mechanism?
Tell me now, why does I know I asked again, but why does genetic bottleneck occur in the first place? Like, what's the thing that limits how much we get in the end?
The genetic bottleneck occurs during oogenesis because only a small subset of the mother's mitochondrial DNA, mtDNA, copies, typically a few hundred out of thousands, is randomly selected and replicated in the developing egg, limiting the mtDNA pool passed to the embryo. This restriction happens due to a natural reduction in mitochondrial numbers in early germ cells before amplification, which doesn't fully represent the mother's mtDNA diversity. This process amplifies the proportion of mutated mtDNA in some eggs, leading to variable disease severity in offspring. Want more on how this impacts specific mitochondrial diseases?
Well, I want to move and talk a bit about mitochondrial myopathies. So, tell me about that. Um,
Mitochondrial myopathies are disorders caused by defective mitochondrial DNA or nuclear DNA, disrupting the electron transport chain and reducing ATP production, leading to muscle weakness, fatigue, and exercise intolerance. They often result from mutations in genes encoding ETC proteins, with symptoms varying due to heteroplasmy and high energy demands of muscles. Common examples include MELIS, mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes, and MERF, myoclonus epilepsy with ragged red fibers, often inherited maternally. Want to focus on a specific type or symptom?
let's talk about LHON and MERRF.
Leber's hereditary optic neuropathy, LHON, and myoclonus epilepsy with ragged red fibers, MERF, are mitochondrial myopathies caused by mtDNA mutations, disrupting the electron transport chain and ATP production. LHON, linked to mutations like MTND4, primarily causes vision loss due to optic nerve damage, while MERF, often from MTTK mutations, leads to muscle jerks, seizures, and muscle fibers with abnormal mitochondria, ragged red fibers. Both show variable severity due to heteroplasmy, with high-energy tissues like eyes and muscles most affected. Want to dive into their symptoms or treatments?
I want to get into their names for the first because I don't get them. Like Leber's hereditary optic neuropathy and myoclonus epilepsy with ragged red fiber. Fuck yeah.
Leber's hereditary optic neuropathy, LHON, is named after Theodor Leber, who described it, with hereditary indicating maternal mtDNA inheritance, and optic neuropathy pointing to optic nerve damage causing vision loss. Myoclonus epilepsy with ragged red fibers, MERF, describes its key symptoms: myoclonus, muscle jerks, epilepsy, seizures, and ragged red fibers.
So if I had to remember like a couple of words for each one of what problems they cause, what would I remember?
For LHON, remember vision loss and optic nerve. For MERF, recall muscle jerks and seizures. These capture the main issues each mitochondrial disease causes. Want to dive into specific treatments or genetic causes?
Let's talk about MELAS now and KSS.
MELAS, mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, causes brain and muscle issues, lactic acidosis, and stroke-like episodes, often due to mtDNA mutations like MTT1, with symptoms like seizures and weakness. Kearns-Sayre syndrome, KSS, involves early onset before 20, eye muscle weakness, pigmentary retinopathy, and heart block, caused by large mtDNA deletions. Both are heteroplasmic, with variable severity based on mutated mtDNA levels. Want to dig into symptoms or treatments?
What is Kearns-Sayre syndrome?
Kearns-Sayre syndrome, KSS, is a rare mitochondrial disorder caused by large mitochondrial DNA deletions, leading to symptoms like progressive eye muscle weakness, ophthalmoplegia, pigmentary retinopathy, and heart conduction issues, heart block, typically starting before age 20. It affects high-energy tissues like muscles, brain, and heart, causing variable symptoms due to heteroplasmy, where the proportion of mutated mtDNA determines severity. Additional symptoms may include hearing loss, diabetes, and ataxia, with no cure but treatments like pacemakers or supplements to manage symptoms. Want more on its management or genetic basis?
I want to talk a bit about treatments now. Tell us about diagnosis, about prognosis. What's the difference between prognosis and diagnosis?
Diagnosis of mitochondrial diseases involves genetic testing for MT.
Keep going.
Super geil, oder?
No, never speak in another language. Only in English, please.
Alright, back to English, no problem. Want to keep going with mitochondrial disease treatments and diagnosis?
Yeah.
Let's keep the energy up. You wanted to talk about diagnosis, prognosis, and treatments for mitochondrial diseases. Should we dive back into that?
Yes. In 2 sentences. Come on. Get on with it. We have 5 minutes left. 2 sentences.
Diagnosis of mitochondrial diseases involves genetic testing for mtDNA or nuclear DNA mutations, and muscle biopsies showing abnormal mitochondria, while prognosis varies widely based on mutation severity and affected tissues, often predicting progressive symptoms like muscle weakness or organ failure. Treatments focus on symptom management with supplements like CoQ10, vitamins, and therapies like pacemakers for KSS heart issues, but there's no cure, and outcomes depend on the disease's impact on energy production. Want a quick dive into a specific disease's treatment?
And how do we prevent? I mean, talk to me about IVF strategy, replacing defective mitochondria.
Mitochondrial replacement therapy, often used in IVF, swaps defective maternal mitochondria with healthy donor mitochondria to prevent mitochondrial diseases, done by transferring the mother's nuclear DNA into a donor egg with healthy mitochondria before fertilization. This technique, like pronuclear or spindle transfer, reduces the risk of passing on mutated mtDNA, but is complex, ethically debated, and not widely available. It aims to ensure offspring have functional mitochondria while keeping the mother's nuclear genetic identity. Want more on the procedure or its challenges?
So how would we do it? We would merge DNA from two eggs, one from the mother with defective mitochondria, the other from a healthy donor, and they will get replaced. But how we know it's going to work?
Mitochondrial replacement therapy involves transferring the mother's nuclear DNA into a donor egg with healthy mitochondria, either by swapping the pronucleus, nuclear DNA, post-fertilization, or the spindle, nuclear DNA before fertilization, then fertilizing the egg to create an embryo with the mother's nuclear genes and donor's healthy mitochondria. Success is assessed by genetic testing to confirm low levels of defective maternal mitochondrial mtDNA carryover and monitoring embryo development to ensure normal mitochondrial function. While promising, it's not guaranteed, as some maternal mtDNA might persist, and long-term outcomes depend on factors like heteroplasmy levels and tissue-specific energy needs. Want more on risk or success rates?
That's all good. Okay, now I want us to talk about an alternative strategy, maternal spindle transfer. What's that?
Maternal spindle transfer is a mitochondrial replacement technique where the mother's nuclear DNA, organized in a spindle structure during cell division, is removed from her egg with defective mitochondria and transferred into a donor egg with healthy mitochondria that has had its own nucleus removed. This reconstructed egg, containing the mother's nuclear DNA and donor's healthy mitochondria, is then fertilized to create an embryo free of mitochondrial disease mutations. It's a preventive strategy for maternal mitochondrial diseases, but success depends on minimizing carryover of defective mtDNA and ensuring embryo viability. Want to dive into how it compares to other methods like pronuclear transfer?
Can you very simply explain what it is? I don't get it.
Maternal spindle transfer takes the mom's nuclear DNA from her egg with bad mitochondria and puts it into a donor egg with healthy mitochondria, which is then fertilized to make a baby without mitochondrial diseases.
But I thought that was the other one that we were talking about, the IVF strategy.
You're right, my bad. For the mix-up, maternal spindle transfer and the broader IVF mitochondrial replacement strategy are related but distinct. Maternal spindle transfer specifically involves removing the spindle, nuclear DNA from the mother's egg with defective mitochondria and transferring it to a donor egg with healthy mitochondria, which is then fertilized. It's one type of mitochondrial replacement, different from pronuclear transfer, which swaps nuclear DNA after fertilization.
Okay.
But both.
Now, I want you to give us one sentence and explain to us if one person had to take away only one thing from today's conversation, what would it be? And honestly, please speak nice and slow.
All right, speaking nice and slow. The key takeaway is that mitochondrial diseases caused by faulty mitochondrial DNA disrupt energy production and can be prevented with techniques like maternal spindle transfer, which uses healthy donor mitochondria to avoid passing on these disorders. Want to wrap up with anything else before we close?
That's great. Thank you so much. Well, guys, thank you for watching. I hope you enjoy these 30-minute videos more than the long ones. And peace.